Now that you understand what your amino acids are, we will now level up and go to proteins. Again, amino acids are the monomers of your proteins. And we will link the properties of your amino acids to the structure, hence the function of our proteins. You have seen this animation. This is a protein molecule that is involved in the transport of small molecules across the microtubules inside the cell. So again, we see in this animation that the structure of a protein is related to its function. So for this protein, its long, slender structure allows one end of it to attach to the molecule it is transporting, and the other end allows this protein to walk along the microtubules. So in this series of videos, you will be encountering a lot of models of your protein structures. You will see this ribbon structure, and you will also see space filling models of your proteins. It is very advantageous if you understand how to look at this type of models. Now, how are your proteins synthesized from your monomers, the amino acids? So the process is basically easy, and it's called a dehydration reaction. If you have one amino acid and another amino acid, so the carboxylic acid functional group and the amine group of the second amino acid condenses. So it's a condensation reaction because the two molecules come together. In the process, one molecule of water is released. And we form what we call a peptide bond, which is highlighted in yellow here. Now, the reverse of the reaction can also take place. The reverse is called a hydrolysis reaction. It's because a molecule of water needs to be hydrolyzed, so the molecule of water is broken down in order to separate or destroy the peptide bond. Now, as you form this chain of amino acids, you can form what we call dipeptide. And when we say dipeptide, it means it's made up of two amino acids. So there is one peptide bond. If we say tripeptide, it's made up of three amino acids. Therefore, you find two peptide bonds, and so on. Now, there are other terms that describe or refer to such chains of amino acids. We also have what we call oligopeptide. There is actually no specific quantifier, okay, as to what an oligopeptide is. So many sources would say that an oligopeptide would be composed of 20 to 25 amino acids. Again, the definition is very loose, okay? Then we also have polypeptide, which some people interchangeably use with protein. Although there's a textbook that says for those peptide chains with a molecular weight of 10,000 Daltons or below, they are polypeptides. But if you have a higher molecular weight, higher than 10,000, then we classify them as protein. But again, the definition is very loose. So this is an example of a penta peptide. So penta, meaning you have 5 amino acids and 4 peptide bonds connecting these amino acids. Now, it's not often used, the term amino acids, but we refer to them as amino acid residues. So the term residue there means that it is what is left behind of an amino acid when it becomes part of a peptide chain. So keep in mind that your peptides have directionality. When they are synthesized, it's the amino end that is synthesized first as it goes toward the carboxyl terminal end. So, the convention when we write the sequence, the amino acid sequence of peptides, is we start from the amino terminal, or what we call N terminal, to the carboxyl terminal, or the C terminal. Now, for short peptides, very short peptides, sometimes people would often write the name of the peptide. So, how do we write the name of the peptide? So, we need to identify the amino acid residues. So, we have here serine. This is glycine. This is tyrosine. This is alanine. And this is leucine. So the convention is to convert the in ending to il and then simply connect the names together. So this peptide will have a name: serine, glycine, tyrosyl, alanine, leucine. So the last amino acid residue, we retain its name. So of course, this is not practical if you have a very long peptide. Sometimes they use the abbreviations. So we write Sir, Gly, TYR, Ala, Liu. And of course, we have the symbols for the amino acids. So again, there is directionality. You always start with the N terminal. So we write this as S, G, Y, A, L.